will be presenting her dissertation proposal today, and her title is A Novel Computational Framework for Inferring the Developmental and Cellular Origins of Rare and Congenital Disorders Using Single-Cell RNA and Sequencing Data. And just a little background about Jennifer. She is a PhD candidate in our BCB program on the National Library of Medicine Fellowship. She is dedicated to uncovering the biological mechanisms of rare and congenital disorders, including her own condition, AMC. During her early years at OHSU, Jennifer found herself drawn to single-cell RNA sequencing data and its utility in studying developmental biology. She is now excited to combine single-cell analysis with systems biology approaches to get to the root of congenital and rare disorders. I should also comment that she last June won the best paper presentation at the NLM trainee meeting. So congratulations and welcome, Jennifer.
Thank you, Karen. Thank you for that very nice introduction. Thank you all for attending my dissertation proposal defense. As Karen stated, I will be proposing a novel computational framework for inferring the developmental and cellular origins of rare and congenital disorders using single-cell RNA-seq data. One second. Okay. So I will begin by providing some background on approaches for understanding the, I'm sorry, my screen is doing something weird. One second. Okay.
It's coming through on our end and it looks just fine.
Okay, thanks. Yeah, I think it's working now. So I will begin by providing some background on approaches for understanding the etiology of disorders and on opportunities for improvement, excuse me. I will then present our proposed project, starting with our specific aims. I will then discuss various aspects of our research approach. I will also talk about the innovation of our project. And finally, I will touch on future directions and give my concluding statements. So let's get started with the research question and what the state of the field looks like so far. 263 to 446 million people, 3.5% to 6% of the global population, are affected by a rare disorder. Yet over 90% of the estimated 7,000 rare disorders still do not have any FDA-approved treatment. Congenital disorders affect over 5 million people born each year worldwide, are the leading cause of infant deaths, and frequently result in lifelong disability. Yet the cause of most congenital disorders is still unknown. These two patient populations exemplify a strong need for research into disease etiology that can inform ways to develop treatments. In many cases, efforts to uncover the etiology of these disorders has been impeded by the limited availability of patient samples, either due to the small number of patients with a given disorder or to the inaccessibility of the relevant tissues during the initial stages of the disease process. Therefore, for these disorders and for others with similar challenges, we need new approaches for determining their pathogenesis that do not rely on the collection of disease-specific data. One important question that still needs to be answered for many of these disorders is which cell types are implicated in initiating the disease process. In other words, what is their root cause at the cellular level? When considering Mendelian disorders, for example, the vast majority are believed to manifest in one or just a few tissues. However, in many cases, we don't know which cell types within those tissues are actually driving the disorder. In addition, many disorders are caused by cellular disruptions that originate before birth. Therefore, another crucial question is what stage of development these cells are in when their functioning is disrupted. Given this observation that disorders manifest in a small number of tissues, a natural assumption might be that the gene responsible for a given disorder is only expressed in those affected tissues or is expressed at a much higher level than in other tissues. Although this is the case for some disease genes, it turns out that this is not the whole story. In fact, most genes are expressed in at least 10 tissues, and an estimated 72% of these genes are expressed at similar levels in disease-associated and non-disease-associated tissues. So this means that we really need to look at more than just the expression level of a disease gene to determine which cell types play a role in the development of each disorder. In fact, there are several reasons why a mutated gene may initiate a disease process in one cell type but not another. For our project, we will be focusing on the mechanisms involving disruptions of the molecular networks specific to each cell type. These will include both cell autonomous or cell intrinsic networks and non-cell autonomous or cell extrinsic networks. In particular, I will concentrate on this concept of gene module integrity, specifically modules of disease genes, to identify the cells and developmental stages implicated in various disorders. So let's talk about disease modules. Barabasi, Gulbase, and Loscalzo defined a disease module as a group of network components that together contribute to a cellular function whose disruption results in a particular disease phenotype. In other words, a disease module represents the concept that proteins and other molecules associated with a disease phenotype are not scattered randomly throughout the network, but instead cluster into modules. It is not the impact of a mutation on a single gene, but the impact of a mutated gene on this entire module that produces the disease phenotype. This idea of a disease module was based on observations from various studies, such as that performed by Go et al., who overlapped a disease gene network with a protein-protein interaction network and saw 10 times more physical interactions between proteins associated with the same disorder than would be expected by chance. Since then, many studies have demonstrated the feasibility of identifying disease modules. One notable study performed by Menche et al. Revealed that out of nearly 300 disorders investigated, 226 were associated with sets of disease genes that clustered together into a neighborhood of the molecular network, as shown here for multiple sclerosis. However, the network used by Menche et al. And by many other studies is a generic network, meaning it is not specific to a given tissue or cell type. If we are to identify the cell types involved in producing disease, we will need to discover what the molecular networks look like within these individual cell types. A few studies have made steps toward uncovering these cell type-specific networks, starting with Kitzok et al. By inferring tissue-specific networks, they were able to identify an average of just 2 to 3 tissues implicated in half of the disorders they investigated. With the growing availability of single-cell datasets, there is now opportunity to identify the individual cell types that initiate disease. For example, Mohammadi et al. Used their method, Skynet, to infer cell type-specific protein-protein interaction networks, and used these networks to infer the cell types that initiated 3 classes of brain disorders. What they found was that there was a clear distinction between the disease classes based on which cell types were targeted, with psychiatric disorders manifesting primarily in neuronal cell types, and neurodegenerative and neoplastic disorders manifesting primarily in glial cell types. A similar analysis on autoimmune disorders revealed disease modules in blood cell types but not brain cell types, again confirming what would be expected. These studies demonstrate the apparent feasibility of identifying pathogenic cell types through disease module detection. However, a number of gaps still need to be addressed to improve the inference of these cell types. The first gap is that the networks used for these analyses are restricted to interactions that have been experimentally determined. This set of known interactions is likely missing a large proportion of the true interactions taking place. and these incomplete interactomes may not provide adequate information to discover disease modules. In addition, these known interactions are biased toward well-studied genes and proteins, meaning these molecules are more likely to form hubs in the constructed network, simply due to the larger number of studies focused on them. This bias can also affect the accuracy of the inferred networks and hinder the discovery of disease modules. The second gap is the need for stronger evidence to support these inferred cell-type specific interactions. Previous studies have relied on the assumption that as long as both neighbors in the network are expressed above a certain level, they must interact. However, as this figure illustrates, there are a number of biological reasons why this assumption is not always valid. Therefore, other strategies that provide stronger evidence of the inferred interactions could improve the accuracy of the networks and the inference of disease modules and pathogenic cell types. The third gap that remains unfilled is the need for a method that can infer pairs of cell types whose disrupted communication results in disease. Communication between cells is crucial for the proper development, maintenance, and function of cell types, and disruption of specific communication activities can result in specific phenotypes. While methods do exist for the inference of cell-cell communication, they do not specifically identify which communicating cell types are relevant for each phenotype. So far, no study that is specifically designed for the inference of pathogenic cell types has addressed this issue. The fourth gap that should be addressed is the need for a pathogenic cell type inference method that can infer multiple disease modules in individual cell types. A single phenotype can result from multiple molecular processes, even within a single cell type. Therefore, its genes may form multiple modules in each affected cell type. So far, studies have evaluated the connectivity of either all of the disease genes mapped to the network or only the disease genes that form a connected component. In either case, the power to detect significant modules of disease genes is lower than if the separate modules were each inferred and assessed individually. Finally, the fifth gap is the need for a method to infer the developmental stages at which the pathogenic cells are most likely affected. Knowing these developmental stages is critical for understanding the etiology of a disease, as experimental studies can be targeted to these periods of sensitivity. In addition, this knowledge can inform the development of treatments, indicating both the necessary timing of treatment administration, such as in utero or ex utero, and the nature of the treatment itself. The approach that we propose will address each of these opportunities for improvement. So let's get into our proposed project, starting with our objective, hypothesis, and specific aims. The objective of our proposed project is to provide a novel computational approach to investigate the cellular origins of disease. In other words, our project aims to understand the root cause of diseases at the cellular level. Our hypothesis is that the observed cell type specificity of a phenotype is the result of the disruption of normal molecular processes that are specific to a limited set of cell types at particular developmental stages. The two aims for our project take complementary approaches for identifying pathogenic cell types and developmental stages. Our first aim focuses on developing a novel computational approach for inferring de novo intrinsic processes happening within the cells, while our second aim focuses on developing such an approach to infer the extrinsic communication processes between cells. Now let's dig into the details. First, I will present our proposed computational framework. This figure illustrates our overall workflow. We will develop a dedicated approach to address each of our aims, as illustrated by the steps above in orange for aim 1 and the steps below in blue for aim 2. While the detailed steps of these processes will differ, they will share the same high-level workflow, as they provide complementary information about the pathogenic cell types. The pipelines we develop will require two inputs, single-cell RNA-seq data and a set of disease genes. They will first infer cell fate trajectories, then construct cell type-specific networks. They will then identify modules of disease genes in each of these networks, and they will output the pathogenic cell types at the impacted developmental stages. With this overall picture of the high-level framework, I will go into more detail about each step. Again, our computational framework will require two inputs. One of these inputs will be a single-cell RNA-seq atlas collected from normal, healthy tissues over multiple stages of development. Our motivation for using data from healthy tissues rather than diseased tissues is twofold. First, it eliminates the need for disease-specific data, enabling the use of our approach on a wide array of disorders. Second, healthy tissues provide a window into normal molecular processes without being obstructed by the downstream effects of a disease process. Because the aim of our project is to investigate the root causes of diseases, our computational framework will use this insight into healthy molecular processes to identify which of these are disrupted in the disease state. This table lists examples of large single-cell RNA-seq atlases that provide data for multiple tissues over various developmental stages. Our approach will be designed to use these large multi-tissue atlases, as well as atlases focused more specifically on the development of an individual tissue. The second input will be a set of genes that are causally associated with a particular phenotype. Because most rare and congenital disorders are associated with a single gene, they cannot be used on their own to identify disease modules. By focusing on a phenotype shared by multiple disorders, we can pool all of their genes together. For example, congenital contractures, more formally known as arthrogryposis multiplex congenita, is a phenotype that is seen in over 400 disorders. Most of these disorders appear to be monogenic and therefore cannot be used on their own to identify disease modules. However, this group of disorders is associated with over 400 genes, and this set of genes could form disease modules. With our strategy of investigating phenotypes rather than individual diseases, we eliminate the need to know the causal gene for any single disorder. Therefore, this strategy facilitates the investigation into the etiology even of disorders that remain undiagnosed. To supply the gene set for a particular phenotype, users of our computational framework will have two options. They can either supply their own gene set, or they can select a term from the Human Phenotype Ontology. This hierarchically structured database of over 15,000 phenotype terms has been linked to over 15,000 genes from the Genotype Ontology. Users will be able to select the genes associated with a specific phenotype term, as well as add to or subtract from this set. Our strategy of combining diseases based on phenotypes is just one of multiple strategies, multiple possible strategies for grouping disorders. Because our gene sets will be constructed according to their association with a specific phenotype, we propose the term phenotype module for the modules inferred through our computational framework, rather than disease module, as we believe this to be a more precise and descriptive term. The first step in our computational framework will offer users context to understand the stage at which cells exist when they are impacted relative to the other cell types in the dataset. The position of the impacted cell types in the inferred cell fate trajectories will indicate the cell's putative progenitors and potential cell fates. To infer these trajectories, our workflow will use the Monocle 3 software, as it was designed to reproduce complex branching trajectories that can originate from multiple progenitors. Our workflow will then need to infer the cell type-specific networks for every cell type in the dataset. This is the primary step where the approaches for the two aims diverge, so I will talk about each of their workflows independently. For aim 1, our computational approach will be to infer a de novo gene regulatory network for each cell type. The construction of such a network will rely on discovering related patterns of expression between genes. However, single-cell data is notoriously noisy, and cells of the same type have relatively similar transcriptional profiles. Therefore, the signal of the true regulatory activities playing out in an individual cell type will likely be masked by the noisiness of the data. Therefore, rather than directly inferring a gene regulatory network for each cell type, we will use a more robust approach. First, our approach will be to learn all of the regulatory patterns that characterize the entire dataset, resulting in a generic gene regulatory network. Then, our computational pipeline will use this learned network as the starting structure from which to infer each cell type specific network. The workflow that we have developed for inferring these networks is based on that of the popular pathogenic workflow. As I discuss our workflow, I will point out how and why it differs from pathogenic. To obtain the generic gene regulatory network for the full dataset, we will first infer an initial network of potential regulatory relationships using a method called GRN-boost2. This initial step simply identifies correlated expression patterns between all of the genes. Therefore, this first step will produce a densely connected network with many false edges, and we will need to refine this network to isolate the true regulatory relationships. In this illustration, the thin arrows represent true interactions inferred by GRN-boost2, whereas the bold arrows represent false edges. When inferring the regulatory network, we need to find the interactions that represent the direct influence of a transcription factor on its target gene. In other words, we want to eliminate any link between transcription factor and gene that is mediated by at least one other transcription factor. Therefore, we will use cis-target to identify these direct interactions. For a transcription factor to directly regulate a gene, it must be able to bind to the gene's cis-regulatory elements. Cis-target makes use of sequence motif data to remove any of a transcription factor's inferred targets whose cis-regulatory elements are not sufficiently enriched for the transcription factor's binding motif. In this illustration, the red arrows represent inferred transcription factor target gene relationships that have low likelihood of direct interaction due to the low enrichment of the transcription factor's binding motifs. These edges will be removed in this step. The remaining false edges are now colored red. Each edge represents a correlated transcription factor and target gene, where the target gene's cis-regulatory elements are enriched for the transcription factor's binding motifs. This means that, in theory, the transcription factor could directly regulate the target gene. However, whether this regulation actually happens within the cells in the dataset is still in question. One way to eliminate correlations that do not represent true causation is to investigate the relative timing of expression for both transcription factor and putative target gene. When given RNA velocity calculations, Scribe can infer the degree to which one gene's expression level over time provides information about another gene's expression level at subsequent time points. Therefore, we will use Scribe along with SC-Velo to infer the putative causal regulatory interactions and eliminate any edges that are not well supported. While the first two steps in our workflow are also implemented in the pathogenic workflow, this step to infer the putative causal relationships is an addition that we believe will further improve the inference of the generic gene regulatory network. By following these three steps, we expect to construct a network that is representative of the direct causal regulatory relationships present in the full dataset. The next task will be to infer the interactions within this generic gene regulatory network that are active in each individual cell type. To infer each cell type specific gene regulatory network, we will first infer which regulons from the full network are active in the given cell type using the AU cell enrichment method implemented in Piscenic. Rather than simply inferring individual interactions based on the expression levels of transcription factor and target gene, AU cell provides greater support for each inferred interaction. This is because it uses the fact that a regulon represents a group of co-regulated genes, and therefore the target genes in an activated regulon should all be upregulated at the same time. The active regulons that are inferred through this enrichment step may not be connected to one another. Therefore, our workflow will also use these regulons as anchors for random walks through the full network in order to infer the paths between them. To guide these random walks based on cell type specificity, our pipeline will weight the genes in the network by their differential expression compared to all other cell types in the dataset. At the end of step 2, we should have a connected set of regulons for each cell type in the dataset that represents that cell type's specific gene regulatory network. So now let's go through the network construction step for AIM 2. In this case, we will be inferring a cell-cell communication network for each pair of cell types. Again, we will break this network inference step down into 2 tasks. In the first task, we will again construct a generic network, this time by integrating prior knowledge of protein-protein interactions. We will then use this network as the basis to infer the cell-cell communication network for every pair of cell types. To form the scaffold for cell pair-specific communication networks, our pipeline will pull the protein-protein interactions stored in the OmniPath knowledge base. OmniPath provides over 100,000 protein-protein interactions, including over 14,000 interactions between intercellular signaling molecules, which are referred to as transmitters and receivers. From the interactions in OmniPath, we will construct a generic cell-cell communication network that contains transmitter-receiver pairs and their downstream signaling pathways. In addition, because proteins upstream of the transmitters and receivers can influence their activity, we will also integrate these upstream protein-protein interactions. Once this generic communication network is constructed, we can begin to infer the communication network specific to each cell type pair. The first step in inferring the communication network for a particular pair of cell types will be to narrow down the initial set of transmitter-receiver pairs. We will be using CellChat, which incorporates information about both protein complex formation and agonists and antagonists to refine its predictions. For example, if a receiver must form a complex to function, but one of the components of the complex is not expressed, the receiver and its associated transmitter will not be included in the network. The next step is to infer the downstream signaling pathways. We will infer these pathways using NicheNet. NicheNet uses an optimized random walk with restart to traverse signaling pathways, beginning at each transmitter, traversing through the bound receiver, signaling proteins, transcription factors, and ending at the regulated target genes. Based on these random walks, NicheNet infers the putative downstream targets of a cell-cell signaling event, and in the process, the putative signaling pathways that mediate this regulation. Thus, we will use NicheNet's predictions to retain the most likely signaling pathways and to filter out the rest. At this point, we can use these inferred intracellular signaling pathways to further refine the network of transmitter-receiver pairs. This time, we will start with the target genes. These genes are predicted by NicheNet to be actively regulated in the receiver cell type. Therefore, we will compare the target genes in each of these regulons with the genes that are actually differentially expressed in this cell type compared to all others. If a predicted set of target genes has low correlation with the actual differentially expressed genes, its associated signaling pathway will be removed. On the other hand, if another regulon's predicted genes have a strong correlation with the cell type's differentially expressed genes, that will provide a high level of support for retaining the entire associated pathway, including the initiating transmitter-receiver pair. So finally, we will infer the upstream protein-protein interactions. Because each gene has a different baseline level of expression, we will create a baseline distribution of average expression values for each gene by randomly sampling subsets of cells. We will then compare each gene's expression level in each cell type to its baseline distribution to determine its statistical significance. Any gene with expression below a significance threshold will be filtered out of the network. At the end of step 2, we should have a cell-cell communication network for every pair of cell types. With the networks constructed, we can then move on to step 3, where our computational framework will infer the modules of phenotype-associated genes. This step will require 3 subtasks: mapping the phenotype-associated genes onto the network, searching for phenotype modules, and evaluating the statistical significance of each phenotype module. The first step is to map the genes associated with a particular phenotype onto each cell type specific network. These genes will be assigned a higher weight than the other genes, which will help to guide the detection of phenotype modules in the next step. Because the phenotype may arise from multiple molecular processes within the same cell type, our pipeline will search for the optimal partitioning of the disease genes mapped to each network. To determine this partitioning, we'll be using a module inference method called hierarchical hotnet. We selected this method for 2 reasons. First, it is able to use the higher weights of the disease genes to guide its partitioning procedure. And second, it explores different hierarchical levels of network resolution, which has been shown to improve the inference of modules of disease genes. With the disease genes partitioned, we can now evaluate the significance of the connectedness and size of each module. For AIM-1, the connectivity of each module will be assessed based on the average shortest distance between the disease genes themselves. In AIM-2, however, we specifically want to identify the phenotype modules that impact cell-cell communication. Therefore, our approach for AIM-2 will be to assess the connectivity of the phenotype modules in relation to the transmitters and receivers in the cell-cell communication network. At the end of our computational framework, we expect to see a subset of cell types at particular stages of development that can be associated with the phenotype of interest based on the presence of statistically significant phenotype modules. Since our second aim is devoted to inferring communication relationships, the cell types will be output in pairs, indicating the necessity of communication between these cell types to prevent the development of the phenotype. Most of the methods we'll be using have adjustable settings that need to be optimized for our purpose of pathogenic cell type inference. These are the hyperparameters of our pipelines, and I will now talk about how we plan to optimize these hyperparameters. As I highlighted at the beginning of my talk, the number of phenotypes for which we know the causal cell types and disrupted developmental stages is still limited. Therefore, to make sure we have enough data to optimize the hyperparameters of our pipelines, we will be using simulated datasets. For our project, there are 2 elements that need to be simulated: single-cell RNA-seq atlases and sets of disease genes for a given phenotype. Oops. Sorry, Sorry, it's frozen. Is it changing on your end? Oh, here we go. Okay. I don't know what happened. Okay. To simulate these single-cell RNA-seq atlases, we will be using a simulation method called Boole ODE. This method is based on GeneNetWeaver, which was used to generate the synthetic networks for the DREAM3, DREAM4, and DREAM5 challenges for transcriptional network inference. These methods begin with a known gene regulatory network from which they sample subnetworks. They then use these known subnetworks, along with a set of differential equations, to model transcriptional events in each cell. From these models, these methods produce a data set of cells with simulated expression values. We will cluster these simulated cells using the popular Leiden clustering algorithm. The next step is to simulate the sets of disease genes associated with hypothetical phenotypes. These simulated gene sets will need to be selected in such a way that they will form phenotype modules in a subset of cell clusters. Therefore, we will select a small subset of simulated cell types in which to construct hypothetical phenotype modules. For AIM-1, the genes in each phenotype module should cluster together in the gene regulatory network. Therefore, we will construct each phenotype module by identifying a connected group of genes in one of the simulated gene regulatory subnetworks and assigning these genes to that module. For AIM-2, we will follow a similar procedure. However, in this case, rather than searching for phenotype modules in gene regulatory networks, our pipeline will need to search for phenotype modules in cell-cell communication networks. Therefore, to construct the hypothetical phenotype modules, we will collect sets of genes known to participate in real cell-cell communication pathways. We will then simulate higher transcription of these genes in the pathogenic cell clusters. To optimize our pipeline's hyperparameters, we will test multiple combinations of values on our simulated datasets. The performance using each combination of hyperparameters will be evaluated based on our pipeline's ability to identify the correct pathogenic cell clusters, as determined by the area under the precision recall curve. Once the settings for our computational pipelines have been determined, we will assess their final performance on a new dataset. This time, we will test our approach on real phenotypes. On the left are our criteria for selecting phenotypes to use in our evaluation. The phenotypes we select will have well-understood etiology with a core set of causal cell types and molecular processes that are activated at known stages of development. In addition, they will have a set of associated disease genes that are enriched for these molecular pathways. Finally, the pathogenic cell types and developmental stages must be represented in available datasets. The table on the right shows examples of phenotypes selected as candidates for evaluating our approach. When considering the datasets we will be using to assess performance, we must remember that our objective is to pinpoint the earliest processes that trigger the initiation of a phenotype. The process of development is what sets the stage for normal physiological functioning, and most of this development occurs in utero. Therefore, even if a phenotype doesn't present itself until childhood or adulthood, the molecular processes underlying that phenotype are typically already established, usually before birth. To detect these early developmental processes, we will primarily use datasets sampled during in utero development, such as these datasets from mouse embryonic development and human fetal development. Using the selected phenotypes and single-cell datasets, we will evaluate our pipeline's abilities to correctly identify the phenotype's known pathogenic cell types and developmental stages. We will also assess their ability to rank their associated cell types and stages above others. Along with this direct evaluation, we will assess additional criteria to support our pipeline's ability to uncover meaningful biology. Specifically, we will assess the enrichment of phenotype modules for known molecular pathways, and for disease genes held out of the mapped gene set. To evaluate the robustness of our approach, we will test its performance on different datasets, such as human and mouse datasets, or multi-tissue and tissue-specific datasets. For each of these assessment approaches, we will compare our approach to that used in the Skynet paper. We expect that our method will be able to identify additional regulatory and cell-cell communication relationships that will improve the inference of implicated cell types and developmental stages. Upon evaluating our pipeline's performance, we may find that we need to try alternative approaches. I will discuss some of the other methods that we will use if our initial approach produces poor results. The success of our pipelines depends on their ability to accurately infer the cell type-specific networks and phenotype modules. Therefore, our alternative approaches focus on inferring these networks and modules in different ways. One approach for AIM-1 will be to infer the generic gene regulatory network using SCODE, which is able to take into account the temporal information that is provided by the inference of the cell fate trajectories. For AIM-2, we will use an alternative method called log-FC-mean for inferring the transmitter-receiver pairs communicating between cell types. This method outperformed all other methods on one of the cell-cell communication tasks in a recent evaluation. For both of our aims, we will also construct the networks through a weighting procedure rather than filtering out interactions with insufficient support. These weights will then be used in detecting phenotype modules. And finally, we will try an alternative module detection method. Unlike hierarchical hotnet, which assumes that the boundaries of the true phenotype modules are best defined by the distribution of the known disease genes, the popular laden clustering algorithm considers only the topology of the network. Therefore, the laden algorithm will not be influenced by our current knowledge of disease genes and any bias that the incomplete gene set may introduce. So now that I have described our research approach, I will highlight the innovative aspects of our proposed project. Our project will address each of the gaps described earlier. Specifically, rather than restricting our generic gene regulatory network to known interactions, we will have developed a novel workflow to infer de novo cell type specific gene regulatory networks from the data itself. This will afford us the opportunity to discover previously unknown regulatory interactions and allow us to dramatically reduce the bias toward well-studied genes. In addition, cell type specific interactions will be inferred using systems-level approaches, reducing the reliance on the assumption that two neighboring genes interact if they are both expressed. We will also have developed a method to infer pairs of pathogenic cell types whose disrupted interaction can produce the given phenotype. To our knowledge, no other study has attempted to identify these relationships. Our pipelines are also the first to use a module detection method to infer multiple phenotype modules in individual cell types. The inference of each individual phenotype module should increase our power to detect significantly connected groups of disease genes. And finally, our pipeline will have built-in functionality for analyzing the temporal component of pathogenesis, enabling investigation of the stages of development at which the relevant disruptions occur. An exciting aspect of our proposed project is that it opens the door to many additional types of studies. I will next talk briefly about potential future directions of our work. The purpose of our project is to generate new hypotheses about the cellular origins of disease. Therefore, a clear research direction would be to experimentally investigate the results of our approach for various phenotypes. In addition, our project can be a stepping stone for other types of computational analyses, such as the development of a disease gene prediction method informed by the networks of the predicted cell types and stages, the integration of our networks and phenotype modules with environmental exposure data to investigate environmental disease mechanisms, and the prediction of downstream effects of the perturbed cell types on cellular development and function. So I have talked about the need to understand the etiology of rare and congenital disorders, and I have proposed a novel project to identify the root causes of disease at the cellular level. I'll now wrap up with some final thoughts. At the completion of this project, we will have provided a computational approach for the inference of pathogenic cell types and developmental stages. We anticipate that our approach will provide complementary information about the connections between the phenotype-associated genes, and we plan to integrate the de novo cell type-specific gene regulatory networks with the cell-cell communication networks to assess whether these complementary relationships can improve the inference of pathogenic cell types. Our project provides an opportunity to study disease etiology for rare, congenital, and other disorders without requiring inaccessible tissue samples from large patient cohorts. In the long term, the insights provided by this approach should facilitate the development of precision treatments that are targeted to the cell types and molecular processes specifically disrupted in each disorder. This table shows the estimated time to complete the various steps of our project. And I would like to thank the members of my committee for guiding me through this process and offering their feedback and suggestions. And I would also like to thank my fellow students for their feedback and support. And finally, I thank all of you for attending my proposal defense. And with that, I can take any questions.
Thank you, Jennifer. We have about 7-8 minutes, maybe 10 minutes for the larger group to ask questions, and then we will, all of us will leave except for the committee and Jennifer to finish up their discussions. So I have a question.
Okay.
My sense is that this approach then could be applied to other conditions as well. So this is a methodology.
Mm-hmm.
What you're really establishing is new methodology and new approaches.
Yes, correct. Yeah, and I agree that this can go even beyond rare and congenital disorders to more common disorders. I mean, yeah, almost anything. I think, if you were going to study, I think this especially applies to disorders, of development, whether that's in utero or postnatal. But I think it possibly, if you had the right data sets, you could maybe study this in, like infectious diseases or something like that. If you have like an actual data set that, is maybe in some animal model or something where you infect and then you kind of, track the development of the disease after infection as well. So you have this kind of multiple time points. I think this could maybe be applied to that as well.
Okay, thank you.
Mm-hmm.
Maybe take your slide down so we can see if anybody's got their hand up. I
Sure.
can only see 4 images on that.
Gotcha. There we go.
So I'll ask a question, and I too am no expert in this area, but I was thinking as you in the beginning of your talk, you were talking about how oftentimes we build models based on data, based on incomplete data, which certainly those of us on the clinical side deal with that, because for example, we only know about patients what's in the electronic health record, which is somewhat complete of a picture, but not always complete. And actually, if you think about other machine learning methods, even outside of medicine, we build these huge models, but really based on the world as we represent it. So I guess my question to you, without knowing a real lot of the details of your work, is will these new methods give you enough additional data to kind of push the needle on building these disease modules more effectively, and then actually lead to the kind of improvements in our understanding that Karen was alluding to?
Yeah, that's a good question. I think that'll remain to be seen. I'm hoping so. I think, for example, when we're inferring de novo gene regulatory networks, again, that's not something that anyone has done before for this purpose. And so I think, as I was kind of talking about in my presentation, it will offer opportunities to discover other interactions that we don't know about in our prior knowledge. One possibility is to combine this method with the prior knowledge networks, and see if that improves things as well, right? That's sort of what we're doing with, at the end when I talked about combining the inferred de novo gene regulatory networks with the cell-cell communication networks. Those are based on known protein-protein interaction networks. And so we're hoping that, by combining those two, we will be able to discover interactions that are already discoverable from the known networks, and then also new ones that we can discover by our inference methods. Does that answer the question?
Yeah, well, like you say, your work will hopefully give us answers to that.
If there's other questions out there from the larger audience, feel free to unmute and ask a question. I think this was a great presentation. I know I learned quite a bit from watching it. So thank you for being so clear and stepping me through your work and very important work. So at this time, anyone who's part of the committee and the educational team that's going to be staying on, the rest of us can go ahead and step off and have a couple more minutes before your next meeting. Thank you.
Thank you.
Thanks.